Great. Thanks, Susie, for inviting me to speak here. Yes, so as you said, I'll be focusing on our implementation of multidimensional MRI and the diffusion frequency. And so just to set the stage, in my view, one of the biggest challenges in neuroimaging is the heterogeneity of the tissue. So typical voxel contains, tens of millions of cell types, shapes, orientations. We have extracellular features as well. We have proteins, we have vasculature, etc. So everything gets basically compacted into a volume average, and we get a scalar, and that's how we use MRI usually. And we kind of lose a lot of the sensitivity and almost all of the specificity doing that. And so, the term we use, multidimensional MRI, the way we use it is it's been in the works for a few decades already, multiple groups. But the idea is that we are, instead of representing or modeling the tissue with a scalar parameter, we derive distributions non-parametrically. And so with this example, with our pipeline, we obtain the distribution of the diffusion, the frequency-dependent diffusion tensors, R1 and R2. Just to show you a kind of a three-dimensional projection of this distribution in the human cortex, uh, we can look at those axes, and one axis is the isotropic diffusivity, right? So it goes from a lower to higher diffusion length. On the y-axis here, I'm showing you anisotropy, which is like a shape parameter. Of course, we all know what anisotropy is. And on the z-axis here, we're looking at the R2 relaxation, right? So lower values are freely diffusing water. Higher values are probably affected by magnetization transfer. And so with our kind of main goal, I guess, in the field is can we decouple these microenvironments using our MRI techniques, right? And so the question is, can we find some kind of mapping? Maybe it's nonlinear, maybe it's not non-unique, but can we find some kind of mapping between these distributions and microstructural substrates in the tissue? And so this is just to kind of our motivation here. One of the most important things we measure is the diffusion frequency or the diffusion time dependency. And so if we simplify it a bit and we think of the brain as a sponge, right? And maybe water are trapped in these spheres or in these pores. So at the very, very short diffusion time, right? Water are only seeing restriction at the boundaries. So everything else, all the other water molecules are free, basically, right? And so the diffusion profile looks very different than in the case of the long diffusion time, in which all molecules are basically seeing the restriction. So there's almost no free diffusion at all. And so we can imagine that these are vastly different. And in fact, this is how we can encode restriction, right? And so traditionally, the diffusion statistical tenor-like waveforms have a well-defined diffusion time, the big delta. And so with our approach, we don't really use only statistical tenor waveforms. We also use free waveforms, so any arbitrary shapes. And so we don't have a well-defined diffusion time. So we work with frequency. So any time domain waveform can be Fourier transformed into the frequency domain. And you can, of course, we know that time and frequency are invertly proportional. So the long diffusion time corresponds to a low frequency, and the short diffusion time to a high frequency. And of course, the higher frequency, we're looking at bigger structure, and lower frequency, smaller structures. And this is very important to remember. And so with the clinical implementation of this, this has already been running for about 4 years. We have, I'm calling it here the 1.0 protocol. It's the Siemens Prisma implementation, 139 volumes, 40-minute scan time, 2 mm isotropic. And I'm showing you here the waveform time domain. So we're using different waveforms in our protocol. And as you can see, some of them are spherical, some of them are planar, and some of them are linear. But we don't have a well-defined diffusion time. So we're looking at the waveform frequency domain, and you can see how how our frequency content looks like in this protocol. And also, this is, we have 139 volumes, but I told you we're encoding different orientations, different echo times, different repetition times to span sparsely the multidimensional space. But with a clinical scanner, even though it's a Prisma, the frequency domain, the highest frequency we get, these are the centroid frequencies here. So we're talking about maybe 21 hertz, and so these are relatively low frequencies. And so this is a limitation when we're trying to encode and to really probe a restriction in the brain. And so we've kind of pushed this forward a bit with what we call the 1.5 version of the protocol on a Philips implementation. So this time we're using multiband. We're lowering the scan time to 25 minutes, and we're obtaining more volumes. And we also targeted the frequency to obtain higher frequencies, right? So that was an important thing for us. And so if we compare the frequency, sensitivity here, this is across the protocol, and we're showing on the axis here the centroid frequency content. And so you can see in red we have our Prisma protocol, and then with black the Philips protocol. So you can definitely see higher frequencies with the Philips protocol. So this was very encouraging, but again, not nearly enough to really look at white matter microstructure. And so this is where we kind of get into this particular collaboration. I'm going to talk about SEMA-X implementation, but everything that we're doing on SEMA-X is also underway on the Connectome 2.0. And so for this project, we had Danny Park here at MGH, basically reprogramming the sequence from scratch, along with Maximian from University of Rennes University, which we basically took Maxime's, preclinical Bruker scanner sequence implementation and implemented it on a clinical scanner here on the SEMA-X. So in this case, these are just some examples of waveforms. Now we're able to modulate the order of the waveforms. We can do first order, second order modulations, which really increases the spectral power here. And we can do that because of the high gradient. So the issue with clinical scanners, of course, is that we need to wait a long time to obtain a reasonable B value, and so we kind of lose the frequency there. So with this acquisition, we're able to acquire 300 volumes, 27 minutes, again with 2 millimeter isotropic resolution. And so the biggest gain, of course, is in the frequency range. So I showed you this already. This is the frequency distribution. And this is what we have on the SEMA-X, our protocol. Let's call it the 2.0 MDMRI. And so definitely there's a huge gain here towards the higher frequencies. And we're very optimistic as to being able to really probe much finer restriction and diffusion length scales. So we take these distributions, and of course, we can derive a lot of parameters out of them. So this is a big thanks to Ro from my group that's doing the analysis here. And so, like I mentioned, we have these distributions of diffusion tensors, frequency-dependent diffusion tensors, R1, R2. So here I'm showing you some parameters. So these are the first order statistics. So these are just the mean parameters in dimensions of the isotropic diffusivity, microscopic anisotropy, R1 and R2. We can obtain variances of these parameters, covariance of these parameters. Here we see the frequency dependency of the isotropic diffusivity and the anisotropy. At the bottom here, we can also separate, components, contributions based on their size and anisotropy, similar to what the Sandy or Nodi are providing us. So these are all similar information. And of course, these are just a few examples of parameters we can derive. And we're doing much more on this to obtain specificity towards some biological pathologies that we're interested in this project. And so this is on the SEMA-X, and our kind of future is to implement it on a Connectome 2 once it's back online, and we're super excited about this. And so, a huge thanks to Brian Edlow here at MGH, who's basically, made it possible, but also to Susie, to Donsug, to Danny, and Maxime at University of Rennes. And thanks you all for listening.